I shall be discussing an important pathway of carbohydrate metabolism, that is glycolysis. In fact, glycolysis is the first step towards energy production utilizing glucose as a starting substrate. The word glycolysis is derived from a Greek word glykos meaning sweet and lysis meaning splitting. So glycolysis is the splitting or breakdown of 6-carbon sugar, glucose, through a series of enzyme catalyzed reactions into 2 molecules of 3-carbon compound pyruvate. The entire sequence of events of glycolysis results in the release of free energy, and this free energy could be utilized for the synthesis of high energy compounds ATP, adenosine triphosphate, and NADH, nicotinamide adenine dinucleotide. Glycolysis is also called as EMP pathway, named after its discoverers Embedin, Meyerhoff, and Parnas. In fact, glycolysis is the only pathway that takes place in all the cells, and the enzymes required for it are present in the cytosolic fraction of the cell. This glycolysis is comprised of 10 steps or 10 reactions, and out of these 10 steps, 7 are reversible and 3 are irreversible. And it's at these 3 irreversible reactions where regulation or control of glycolysis takes place. These 10 steps of reactions in glycolysis are divided into 2 phases. First is the preparatory phase, which is also called as investing phase. It's called so because here ATP is invested or consumed, and the first 5 reactions of glycolysis are included in this phase. The second is the payoff phase, and the last 5 reactions of glycolysis are included in it, and ATP is produced during this phase of glycolysis. As we know, the starting substrate of glycolysis is glucose, and this glucose needs to enter the cell. Once inside the cell, glucose needs to get activated. This activation is done by phosphorylating glucose at position number 6, forming glucose 6-phosphate. Phosphoryl group donor at this step is ATP, and the enzyme catalyzing this reaction is hexokinase. Next, this glucose 6-phosphate undergoes reversible isomerization to fructose 6-phosphate, and the enzyme catalyzing this reaction is phosphohexose isomerase. This step of reversible isomerization of glucose 6-phosphate, which is an aldose, to fructose 6-phosphate, which is a ketose, is necessary in the overall chemistry of glycolytic pathway, as the rearrangement of carbonyl and hydroxyl groups at carbon at position number 1 and carbon at position number 2 is necessary for the next steps. Next, this fructose 6-phosphate once again undergoes phosphorylation, resulting in the formation of fructose 1,6-bisphosphate. Here again ATP is the phosphoryl group donor, and the enzyme catalyzing this reaction is phosphofructokinase 1. This phosphofructokinase 1 is different from the enzyme phosphofructokinase 2, which catalyzes the phosphorylation of fructose 6-phosphate to fructose 2,6-bisphosphate. Next, this fructose 1,6-bisphosphate undergoes cleavage, and it's this step which gives glycolysis its name, and the enzyme catalyzing this reaction is aldolase, resulting in the cleavage of this 6 carbon phosphorylated sugar to 3 triose phosphates. One is glyceraldehyde 3-phosphate and the other is dihydroxyacetone phosphate. This dihydroxyacetone phosphate reversibly and rapidly gets converted to glyceraldehyde 3-phosphate because it's this glyceraldehyde 3-phosphate which is now degraded in the subsequent steps of glycolysis. So this was the preparatory phase of glycolysis, and here we found that 2 molecules of ATP are consumed, resulting in the formation of 3 carbon compounds, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. In the payoff phase, which starts with glyceraldehyde 3-phosphate, here we shall be considering now 2 molecules of glyceraldehyde 3-phosphate. One molecule which is formed after the cleavage of fructose 1,6-bisphosphate, and another molecule of glyceraldehyde 3-phosphate is formed from dihydroxyacetone phosphate. Next, this glyceraldehyde 3-phosphate undergoes oxidative phosphorylation, resulting in the formation of 1,3-bisphosphoglycerate. And here at this step, phosphorylation is done by inorganic phosphate rather than by ATP. Here also oxidized NAD gets converted to its reduced form, that is NADH, with the release of protons which move into the solution. And the enzyme catalyzing this reaction is glyceraldehyde 3-phosphate dehydrogenase. Next, this 1,3-bisphosphoglycerate gets converted to 3-phosphoglycerate, and the enzyme catalyzing this reaction is phosphoglycerate kinase. This step of glycolysis is the first substrate level phosphorylation. It's called as substrate level phosphorylation because here ATP is formed, and the donor of phosphoryl group at this step is the substrate, which in this case is 1,3-bisphosphoglycerate. Next, this 3-phosphoglycerate gets converted to 2-phosphoglycerate, and the enzyme catalyzing this reaction is phosphoglycerate mutase. This enzyme basically shifts phosphoryl group between carbon at position number 2 and carbon at position number 3. Next, 2-phosphoglycerate gets converted to phosphoenol pyruvate, and the enzyme catalyzing this reaction is enolase. At this step, there is a reversible removal of water molecule. Next, phosphoenol pyruvate gets converted to pyruvate, which is the end product of glycolytic pathway, and the enzyme catalyzing this reaction is pyruvate kinase. This is the second substrate level phosphorylation, as at this step also ATP is formed. So this is the payoff phase of glycolysis, and in this phase we found that 4 molecules of ATP get formed, but the net ATP formation in glycolysis is only 2, as 2 molecules of ATP are already invested in preparatory phase of glycolysis. One other thing is that the energy in the payoff phase is also conserved in the form of 2 molecules of NADH. Next, the end product, that is pyruvate, which is formed in glycolysis, has 3 different fates. First is when the conditions are aerobic. Pyruvate gets oxidized to acetyl group of acetyl coenzyme A. This acetyl group gets converted to carbon dioxide in TCA cycle, which is also called as Krebs cycle. The other fate of pyruvate is when the conditions are anaerobic or hypoxic. It gets reduced to lactate and the process is called as lactic acid fermentation. The third phase of pyruvate is also seen under anaerobic or hypoxic conditions, where this pyruvate gets converted to ethanol and carbon dioxide, and the process is called as ethanol fermentation. So this was all about glycolysis. In the next video, I shall be talking about the regulation of glycolytic pathway.